Hi, everyone. Thank you so much for joining us. And as you just heard, we have two fantastic speakers, and Breeya and I will be hosting the seminar today. Currently, I am a postdoc at Yale School of Medicine, and Breeya is working at Harvard Medical School. And just want to give you an idea of the structure. Each speaker will give 25 minutes presentation, followed by 5 minutes QA. We encourage you to raise your hand during the QA section, or you can type your question in the chat, just to keep in mind that there could be some lag between the system updating. So we really don't want to miss your question. So raise your hand if you can. And there will be a 10 minutes final QA by the end of the two talks. The purpose of this final QA is to really have the opportunity for you to know the two speakers better. You can ask questions that didn't get answered earlier, or you can ask them more general questions regarding to their career development. And this will be a fantastic opportunity to really get to know them well. And let me move this slide. Interesting. Okay, technical issue. Let me, okay, so I wanted to also take the opportunity to promote an upcoming seminar on the neurocontrol of sickness behavior on October 6th. Please mark your calendar if you are interested, and stay tuned for more details. We have three fantastic speakers also lined up for these topics, and mark your calendar. And without further ado, I want to introduce our first speaker, Brittany, who is now an assistant professor at Indiana University of School of Medicine. And Brittany has been an expert of bacteria since graduate school, where she engineered the coding of the bacteria for a variety of benefit, which sounds magical to me. And she did her postdoc training with Sakis Masmanian at Caltech. This is where I meet Brittany. She has this most warm smiling ever that you can see on a person. And she's a fantastic person who you are going to know her very well during her talk. And she is currently hiring. So if you are interested, please hit her up with emails, and I will put her lab website in the chat. And please take away, Brittany. We are looking forward to your work.
Thanks, Vivian. Okay, let me share my screen. All right, if there's any trouble, somebody unmute themselves and let me know. But okay, so like Vivian said, I am really interested in microbes and their surfaces and what they produce and how they signal with the host. And I'll tell you about one molecule in particular today that I've studied as a postdoc mostly, and I'm following up on now. Let me move so the sun is not in my face. And just a little bit of context, of course, you all know that there's this community of microbes that inhabit the gastrointestinal tract. It's made up of a bunch of bacteria, viruses, fungi, archaea, and it's really diverse and really dense. Any one of us could have up to 1,000 bacterial species in our gut. Many of those haven't even been cultured anywhere else in nature. And you can see by these electron micrographs here that the bacteria I'm studying really get quite close up to these intestinal epithelial cells. So it's really, we share a very intimate relationship with this community of microbes. And it's easy to imagine that there's a lot of bidirectional signaling constantly happening at this interface between the microbial community and the host tissue and beyond the gut. And a lot of things can
it would be her, I was
influence the microbial community that we have. Some of those are things we can control, like diet, exercise, other lifestyle choices. We also have a lot of genetic predispositions that influence the microbiota, and this community also does shift quite a bit as we age. Although in general, it is a very stable community of microbes, and what we do affects it, and what it does affects us. So a lot of the signaling that these microbes do and their various activities really are a benefit to the host. They modify and break down dietary components that are complex and provide us vitamins and nutrients, protect us from invading pathogens. We don't get as sick, and they're really crucial for proper development of the immune system and the nervous system. So we know that the gut microbiota is very important and it plays a lot of roles, has a lot of impact. But figuring out all the details and untangling this web from this complex microbial community and our own complex host systems has been a huge endeavor. And one thing that complicates that even more is that there's a lot of redundancy in the microbial population. So you might have two different microbiotas, A and B. They're pictured here, just colored in differently. So maybe the profile is very different in the community, but then the functional readout of roles that the microbes are playing or molecules that get metabolized might be the same, or it could be different. So that adds further to the complexity. So one way around that I like is to take a step away from figuring out what microbe is doing what, when you might have, you remove one and then there's no difference because there are 10 other that could take its functional place. And so I like to look at the microbiota from the functional readout. That's possible because the microbiota encodes, there's over 22 million genes in the microbial population in the human gut, compared to our genome, which is much closer to 22,000. So there are all these unique enzymes that come into contact with host-derived and diet-derived molecules. And these molecules get filtered through this microbial population, and these molecules get transformed and metabolized and modified into a vast pool of molecules, and each one of them could potentially be a signal sent to the host. And just one quick example, so amino acids that maybe don't get absorbed earlier on in the intestinal tract and come into contact with microbes in the small intestine or in the colon can be broken down into neurotransmitters, neurotransmitter precursors, and many other molecules like indoles, polyamines, and phenols. So all of these molecules could potentially play a role in signaling with the host. And these are harmful or beneficial, and I really think many of them may be the major players in the phenotypes that we see in interactions between the gut microbiota and the host. And I also think it's pretty cool as a therapeutic target. So at any level along this pathway, the substrate, the microbes themselves, and the enzymes that they produce, and the microbial byproducts that they're making could all be very tractable therapeutic targets. So in my work, I have focused on trying to resolve some of the mechanisms between the brain and the gut and these signals. And part of that is because there are a lot of neuropsychiatric conditions that have shown to be associated with differences in microbial populations. This is the case in a variety of disorders and conditions, but it's been really difficult to move away from a correlation that there are these populations of patients compared to controls, and they have different microbial communities and profiles, but is that just happenstance, or is it causative in some way? And a lot of people are trying to solve that problem, and a lot of progress has been made. But at the beginning of this work, I was really interested in asking, are there specific signals produced by the gut microbiota that can affect the brain? And what are these, and how is it happening? To move away from the correlational studies where it's hard to tie specific mechanisms. So I've done this in the context of autism spectrum disorder, which is characterized by abnormal social communication and increased repetitive behaviors. And it has various common comorbidities. And one that quickly became at the forefront of my work was anxiety. And this began with my opportunity to analyze metabolomics data from over 200 plasma samples from individuals with ASD compared to controls, and a little under 100 paired fecal samples from these same individuals. And we did as untargeted as we could get metabolomics, and identified a really big group of metabolites. We did this with Metab1. And of all of these over 1,600 plasma metabolites that we were able to identify, bacterially produced metabolite was the one with the highest fold change. And this is written here as 4-EPS, which stands for 4-ethylphenyl sulfate, and it was about 7-fold higher in the population of ASD individuals compared to the neurotypical controls. And when you use machine learning to ask what molecules are the most discriminatory between the ASD and the neurotypical groups, 4-ethylphenyl sulfate is in the top 25 ranked molecules, and that's from all molecules detected in the blood. So it was pretty exciting that it was an interesting bacterial molecule to study. And another reason that they made this something that I wanted to follow up on is that this molecule we also identified in a mouse model of atypical neurodevelopment, and had been previously identified in another mouse model by someone else. Elaine Chiu, who's now at UCLA, found this molecule to be a lot higher in a mouse model she was working with. So we moved forward and engineered bacteria to produce this metabolite, 4-ethylphenyl sulfate. So we confirmed along each step of the pathway that the engineered pathway we were working on does form 4-ethylphenyl sulfate. It's a tyrosine metabolite, and it moves through the pathway, and the bacteria actually only produce 4-ethylphenol, so 4-EP. And then we confirmed that host enzymes are capable of sulfating this group, and it happens pretty rapidly, and the sulfotransferase is ubiquitous in the host. It's in the intestinal tissue and in the liver. It's even in the brain, and so we really can detect mostly in the host this molecule in the 4-EPS form. And this provided an opportunity to be able to take wild type mice. So all the mice are wild type in this whole presentation. And these were germ-free mice that we put either bacteria that could not produce 4-EP or bacteria that could produce 4-EP. So everything else is the same. The bacteria are the same besides knocking out or overexpressing this pathway. And then the mice hang out, and then we test them after they've been exposed to this molecule through their gut bacteria for a while. And we confirmed that 4-EP is made and it's detected in the feces, and that once it gets into circulation, we can detect it as 4-EPS in the urine, in the serum, even in the brain. It seems to be able to cross the blood-brain barrier. And I looked elsewhere in the body, but I didn't see any obvious effects, and quickly focused in on the brain. And one of the kind of broad looks we took to look at the brain was functional ultrasound imaging. And with this method, these mice are anesthetized, but they're alive. And this method takes advantage of the cerebral blood flow. And it's really just like any ultrasound you're familiar with. There's a probe that's extracranial, and you can see the blood flow and the coloring. It's a very cool technique. And here's just a single shot with a map, a brain atlas overlaid. And so we've numbered off arbitrarily brain regions, and then you can correlate the activity in one brain region with another. And I've shown that here as a correlation matrix. So this is just along the bottom, these brain regions, and then those same ones along the top. And then we can correlate the activity with each other. And just by eye, you can see along the bottom in 3 different bregma regions. 4-EP exposed mice have a higher level of functional connectivity in the brain than the mice that were not exposed to 4-EP. And just kind of summarized down here some of the brain regions that are responsible for that change in functional connectivity. And a lot of the regions are associated with the limbic system. So could be playing roles in things that the limbic system controls, like fear or anxiety, among others. And so those brain regions that kind of pop out are the amygdala and the hypothalamus. But to move forward to higher resolution, we performed another brain imaging study using autoradiography, where you inject a radioactive tracer. So similar to scans you might get on your own brain, where they inject something so that it'll show up well. This one's more like a PET scan because it's radioactive, and it gets trapped in metabolically active cells. So it's a proxy for brain activity, and it's just a single time point. And you can see in these images here are summary. So this is all of the 4-EP plus mice over the 4-EP minus mice. And anywhere that it's a warm color, it's higher activity in the 4-EP plus mice. So mice exposed to this molecule from their gut bacteria have elevated activity in the amygdala and hypothalamus and other regions of the brain, and a lot of overlapping regions that we saw with the functional activity assay, even though they're kind of assaying different elements of brain activity. So we were excited that maybe the limbic system and these anxiety and fear circuits might be altered by this metabolite. So we did a whole host of behavior tests. I'll just show you the ones about anxiety. So we did elevated plus maze and open field tests, which both take advantage of kind of mice want to explore, but they're also prey and they are fearful. And so they also are very timid and they hide. And so in these mazes, they tend to stay in the areas where it's darker and more protected. That could be on the edge. This is kind of looking down on a box that's open. And so they spend more time on the edges or in these closed arms. And then a less anxious mouse will spend more time exploring in this open arm or in the center of the box. And we can quantify that. And a more anxious mouse will also dig more and bury more marbles in this marble bearing assay. And so in the presence of 4-AP produced by these bacteria, we see decreased exploration in the exposed regions and increased marble bearing, which is consistent with an elevated anxiety-like behavior. So this metabolite produced in the gut is causing these mice to have more anxiety-like behavior. But we had no idea why or how. So we took the brain regions that seemed to be the most interesting in our brain imaging studies, and we performed quant-seq, which is basically mRNA sequencing. And we observed that one cell type in particular popped out, and that was oligodendrocytes. And we observed that oligodendrocyte precursor cells were, well, genes particular to oligodendrocyte precursor cells were elevated, and those linked with mature oligodendrocytes were downregulated, and that's kind of some representative genes are shown here. So in green, you can see these mature oligodendrocyte genes are lower. Some of these early oligodendrocyte genes are higher. And oligodendrocytes are cool cells. They're, of course, the myelinating cells. Mature oligodendrocytes are producing the myelin and wrapping neuronal axons for proper function. And you can see some grown in vitro here where this myelin is laying right on top of the red axon. These oligodendrocytes really branch out onto multiple axons. These are just pure culture here, or mixed culture. But it's been shown in a couple of studies before that the microbiota might be able to influence these cells. Germ-free mice have thicker myelin in certain brain regions. This is the myelin in gray. And mice on broad spectrum antibiotics wiping out all the microbes also have altered profile of oligodendrocytes. And one more thing that was very cool about this result in these cells is that I feel like a new study is coming out every day showing that oligodendrocytes are playing a lot of roles, not just in insulating axons and providing metabolic support, but also important for motor learning, memory, fear, and various disease models, in addition to simply myelin-linked diseases. And so they're very cool cells, and I think they're really becoming appreciated as having roles in a lot of functions. This is in humans as well, even into adulthood. When we learn new skills or have new experiences, myelin plasticity and myelin remodeling is crucial and very involved. So to follow up a little bit on the results that we saw from our mRNA sequencing, to see if these cells really are influenced rather than just you have to make sure the sequencing data means something. So we looked at the cells themselves, and indeed the 4-AP exposed mice have elevated levels of immature marker of oligodendrocytes and decreased level of a mature marker, and that's quantified here as a ratio. We saw the same thing, the same phenotype by flow cytometry. And then we also wanted to know, okay, the cell differentiation patterns are different. Is that having an effect on the functional readout and the myelin itself? And so this is electron microscopy, and these images. So this is 4-AP plus all down the column on the right, and 4-AP minus mice on the left. And this top row here this is the corpus callosum, where there's lots of axons in parallel, and they're kind of coming out from the plane of the screen, and then we take a cross section, and the darker gray circles are myelin. And so these two images, or these two groups of mice, actually have the same number of axons. We didn't see any differences in number of axons in any of these brain regions. But when we quantify how many of them are encircled by this dark circle, so how many of the axons are myelinated, we saw that the 4AP plus mice, I think it's most visible in this picture probably, have an increased level of unmyelinated axons, and that's quantified here on the left. And these are just higher resolution images that also show you that the myelin level itself is a little bit thinner in the 4AP plus group than the 4AP minus. And I know I'm just showing you a couple of representative images. Here's a longitudinal one along an axon. This is a cross section kind of at the end of an axon. And this was consistent throughout many images, many regions, and many mice. So we feel confident that the myelin is truly different. And the only thing that has changed between these mice is the presence of this gut bacterial metabolite. We confirm this using an MRI technique that allows us to quantify the disorder of myelin. So this is diffusion tensor imaging. And we see that there's more disorder in the 4AP plus mice, so less order in the brain. That's in the whole brain and in various brain regions that we looked at. So it's pretty cool. All of this is a result of this one bacterial metabolite. We knew that the metabolite is produced, we can selectively produce it by microbes, and it gets in the body and it influences behavior, and it influences the brain, and it influences oligodendrocytes. But I wanted to know, are these phenotypes linked to each other? So are oligodendrocytes responsible for the differences that we see in behavior? And I don't think I fully answered that question by any means, but what we have done is I treated the mice with an oligodendrocyte promoting drug. So it pushes the differentiation of oligodendrocytes toward mature myelinating oligodendrocytes. And if you look just at this dark green bar, it's the same group that we've been seeing all along. These are different mice, of course. And then these mice, well, this group of mice, half of them were treated with a drug. This light green is then on the drug. So this is the myelinated ratio. We see that the oligodendrocyte ratio is restored. And then the behavior was restored too. So when they're put on this drug, they start spending more time exploring, similar to controls, and same in the open field test. And then in the marble bearing test, they start digging less and bearing fewer marbles. And so we conclude from this that the oligodendrocyte phenotype is key for these altered behavior phenotypes that we're seeing as a result of the molecule. And I kind of just summarized on accident as I was talking. So we have identified this metabolite and tried to see what's going on in the brain. But I want to tell you one little follow-up study that we were also able to do at the same time. These microbes are producing the metabolite, and we're able to control it. And this metabolite is not produced by the host in any way. So it's strictly bacterial if it's present. And so it's produced in the gut, and then it gets into circulation and influences organs and the brain. And I was interested in asking, what if we use a drug that can absorb the molecule and just target it for excretion, limiting how much gets into circulation and then is exposed to the brain. So we use this oral absorbent. It's really kind of small spheres, almost like activated charcoal, but very specific. And it binds the molecule and it's excreted. And we tested this in mice. And sorry about the color change. This is for a different project kind of. So this orange group, it was the dark green from the slide before. So these are 4AP plus mice. They're exactly the same. They're germ-free and they're colonized with the same microbes as before, compared to the counterpart that doesn't produce 4APS. And so we see no levels, and then it's produced by the bacteria. And then we put them on the drug, and these levels were decreased. And it doesn't get rid of it all the way. We feel okay about that because in our human studies, like in any human study, any human sample I have looked at, we're able to measure some levels of 4APS. So I don't know if that holds true across the entire population, but it does seem to be a very ubiquitous molecule, and perhaps there's just a threshold where it can become problematic. And so we carried through with these mice to see if just simply lowering it is sufficient to alter the behavior. And it was. So the mice on the drug, which is now in blue, showed again an increase in exploratory behavior in these anxiety tests. So they had less anxiety-like behavior, and again, buried fewer marbles on the drug. So it looked like it was working. It was exciting preliminary data, and we were able to work with Axial Biotherapeutics, a biotech company, to do a clinical trial. So it was a phase 1 open label trial with a little bit under 30 teenagers on the autism spectrum. And many of them had comorbid anxiety and GI symptoms, and were ideal for a study looking at kind of a gut-brain axis. And we used the same drug as what we used on the mice. Although in the mice, the drug was incorporated into the food, and in the human study, the clinical study, it's just it's like drinking a very tiny vial of very small, perfectly round nerds that probably don't taste as good. So they were on treatment for about 8 weeks, well, exactly 8 weeks. And then we did various metabolomics and behavior tests throughout the study. And it's pretty cool. It was awesome to be able to be part of a clinical study anyway that was based on my work, and get it moved toward a translational setting and see if it actually matters in humans rather than just in my mice that are very kind of a simplistic setting where it could easily not be translational. So the drug does reduce 4-EPS in these individuals, except for maybe one person. I don't know what's going on there, but overall, a pretty nice reduction of 4-EPS levels in the blood. And then we measured anxiety and irritability. And so this orange is the baseline. Then they're on the drug for 8 weeks, which is in blue, same thing here. And then there was a bounce back washout period where they no longer have the drug for a few weeks, and then they come back. And so on the drug, there's a decrease in anxiety. It's like a clinically significant level, and a decrease in irritability. And that did bounce back some after this washout period once the drug had been removed, which is very exciting. So we're able to see that this metabolite that looked interesting in mouse model and in metabolomics data from some clinical samples could be engineered and studied for its specific influence in a causative way in the brain, and assess the brain in various ways, identify oligodendrocytes as the potential target. I don't know yet if it's the only target, or if it's the first target, or if it's a secondary effect. Those are all questions that I'm working to answer now. We have some indication that it is a direct effect on the brain, and we have some proteins of interest that we think are binding partners with 4-EPS. So I'm really interested in teasing out the mechanism of how this exactly is working. And drugs that were used to induce oligodendrocyte differentiation were sufficient to reverse this effect. And then using a drug in my mice and in a clinical setting was sufficient to keep 4-EPS from entering circulation as much and be excreted, and then improve anxiety and irritability in the teenagers. And so the study is now going to be they're going to do a phase 2 study and move forward and have a placebo group and really try to tease out some of those kind of details. And I've had a lot of collaborators throughout this project. All of them have been super instrumental, help in the engineering and in the brain imaging, and just throughout. None of this could have happened without them. So I've really appreciated working with all of them. And thanks for listening. And I'd be happy to answer any questions. I was talking really fast, I feel like. So definitely let me know if there's anything you want me to go back and go over.
Thank you so much, Brittany. That was fantastic, especially the clinical trials. Looks really exciting. We already have 3 questions lined up in the chat. Let me just bring it up to you. And the first question is whether you have purified the 4-EP itself, the precursor of the 4-EPS, and then treat the drain-free mice with the 4-EP, and what effect did you see? Do you see a similar trend, or it's different compared to without the 4EP?
Yeah, we have done that. So we've administered 4EP and 4EPS. 4EP you can just buy, and then we've had a chemist synthesize the 4EPS. And if I administer that in the drinking water or by injection or by gavage to regular SPF, like conventionally colonized mice, we do see a lot of the same phenotypes. It's not a perfect overlap. There seem to be some, there's some interplay with the other gut microbiota and in an SPF background. But they do have elevated anxiety and some shifts in their oligodendrocyte maturation profiles. I tried putting in my engineered microbes into those SPF mice, but then I couldn't detect the 4EPS. So the other microbes being in there made it complicated. So there's still things to answer.
Yeah, that's exactly my question. That sounds like there's some local processing of the 4EP, but let me just get to the second question. Among the things that you mentioned affect our microbiome, do you know the impacts of this molecular itself, whether it self-regulate the 4EP or 4EPS, whether then self-regulate the composition of microbiome or the question is about whether there's an effect by this molecule itself on microbiome.
Yeah, that's a good question. I don't know. I think that so it's basically a phenolic small molecule. And I hypothesize, and I haven't fully tested yet, that it probably has some bactericidal effects on some microbes. And I think there are some enzymes that break it down for that reason, that might be one of the reasons that bacteria have evolved enzymes to interact with these molecules. And I have some targets I want to test to see if they do actually break it down. And I have put mice on a really high level of 4EPS and then measured the microbiota, and there were some interesting changes. I didn't do metagenomics, I did 16S profiling, and as any of you have done that know, then you just get a bunch of results that are hard to do anything with because they're very general and it's not like at the species or strain level sometimes. And so there are some changes, and I'm interested in following up on that, but I haven't yet, asking like who exactly is producing it. So I've engineered this pathway, like what microbes are actually producing it in a host setting. We've identified that there are more than one pathway to make it, and then what microbes are breaking it down and kind of what's happening in the gut with this molecule. And those are all questions I don't really know the answers yet.
We have one more question. Are different microorganisms present evenly throughout the gut, and meaning including fungi and bacteria, and whether they have spatial location for each specific colonies, and what's your thought on this particular bacteria?
Ask me again if I don't answer the question accurately, but there is a lot of microecology in the gut. So there are specific niches that certain bacteria or fungi or other microbes have evolved to be really good at inhabiting. And there's a lot of competition for different niches and resources in the gut. And in my mice, none of that was happening because these were germ-free, and I only put in the microbes that produce this one molecule or that didn't. And so in the experiments I showed today, none of that competition or interaction with other microbes was happening. I did that on purpose. It's not very realistic, none of us have a single microbe in our guts. I did that to be able to show causation specifically with that one molecule. But yeah, absolutely, in a real setting, which made the clinical trial so exciting to me because it's real humans. There is a lot of competition and specificity to different locations within the gut. I don't know if I answered the question.
If I may ask just quickly one more question. In the animal model, it seems the effect of 4EPS has much greater effect on males versus the females. And did you see that in the ASD patients, or whether the 4EPS level changed, and how does it become higher in the first place in the ASD patient? I was just curious about this part.
Yeah, so we did see a higher effect in the males than the female mice. We did see it in the females also. It just was a lot stronger in the males. And that is, it's tempting to be like, oh, and then in ASD, like the diagnosis rate for males is much higher. Whether or not that's actually a link, or I don't know. But in our clinical study, we had all males. We didn't restrict it. Anyone was allowed to be admitted to the study. It was not based on sex, but this was interrupted by COVID. And so we had to stop enrollment. And then there were only, there's only 1 or 2 females that were enrolled, and one had to drop out because of something unrelated to the study. So anyway, we ended up with just males in our study. And so I don't know, it'd be really interesting. And as far as what makes the levels be higher in ASD, it could be a variety of things. Like it's a bit of a chicken and the egg, right? Does the microbiota contribute to the initial etiology early in life in some of these ASD phenotypes, or is it kind of a feedback loop, or some of the dietary preferences of ASD individuals might affect 4-EPS levels? We tried to control for that as much as we could in both of our metabolome study and in the clinical trial. And then also just other differences in ASD individuals compared to neurotypical controls might be driving these changes that we see in the microbiome. And so I'm really interested in answering some of those questions, but I don't think we know. I do want to say though, I don't think that this molecule is causing autism. I think it's one piece of a very multifactorial system, and that one that luckily we can toggle and influence pretty easily because it's not a host enzyme.
Thank you. Thanks Brittany again for fantastic presentation, and let's welcome Breeya, and she will be telling us the next seminar.